Hello everyone, this is Christine, and today I want to go through the WNT signaling pathway, or we can call it Wnt pathway. So this is one of the signaling pathway that I have to understand for my research about cancer cells. So I just want to go through it and just want to share my knowledge and information. So for the Wnt pathway, this is a really important signaling pathway for cancer cell research. And so we have in the inactivated stage, we have the receptor freehold and LRP are going together. They are transmembrane receptor. So in the absence of Wnt pathway in the inactivated state, we have a complex structure. This is called destruction complex. It's actually the beta-catenin destruction complex. So it doesn't matter how many beta-catenin in our body that we produce, because of the presence of this destruction complex, our beta-catenin is always in a really small concentration, really small level, because it gets destroyed in degradation. So the destruction complex structure, including around like 7 proteins, important proteins. We have DVL, ASIN, CK1, CSK3, APC, beta TRCB, and our protein beta-catenin. So this protein is known as a protein for transcription activation. So in the presence of beta-catenin, the cells will grow and will divide and reproduce. But in the absence of beta-catenin, the cells hardly can reproduce and divide and multiply. So in the inactivated stage, without Wnt, we have this complex structure. We have that this complex structure will phosphorylate the beta-catenin, and also that when beta-catenin is phosphorylated, this will send a signal to beta TRCB, and beta TRCB will ubiquinate beta-catenin. So this beta-catenin is phosphorylated and ubiquinated. This will send a signal to proteasome to come here and destroy beta-catenin into like small pieces. So it is degraded. So this process is degradation of beta-catenin. So this will keep beta-catenin at a really low level in the cells without the presence of Wnt. So in the inactivated stage, beta-catenin is kept at the low level. So because in our nucleus there's an absence of beta-catenin, usually the transcription factor will be repressed by several proteins. And there we have an important protein called Crotrol. So this protein acts as a repressor that is repressed the transcription factor, and so the target genes do not get transcribed and do not get translated. So this is a repressor and there's no target gene transcribed. So in sum, without the presence of Wnt, in the inactivated state, beta-catenin is kept at a low level, and the target gene is not transcribed. However, in the presence of Wnt will come here and it will attack and interact with the receptor, first with freehold receptor. This freehold receptor in turn will phosphorylate the LRP receptor. This LRP receptor is activated and phosphorylated. So in turn, this will induce the destruction complex to come close to the place between freehold and LRP. So when it's come close here, our protein DVL will be activated. When this is activated, it will inhibit the destruction complex from phosphorylating our beta catenin. And also the second function is that keeps the beta TRCB separated from the destruction complex, and this will prevent the beta TRCB from ubiquitinating our beta catenin. So because of these two reasons, that beta catenin is not phosphorylated and it is not ubiquitinated. So beta catenin is kept at a really high level in our cells. In turn, it will be at a really high level in our nucleus. And beta catenin will replace the place of crucial. And in turn, beta catenin acts as an activator for our transcription factor. And this whole complex will go to the target genes and transcribe target genes. So in the activated state, and where there's a Wnt, there's a Wnt signal, then beta catenin will keep at a high level. And in turn, our genes will be activated and all the target genes will be transcribed. These genes can be can lead to the growth of the cells, and also it lead to the divide and multiply of the cells. So cancer happen when one of the protein in our pathway is mutated. For example, if one of the protein in this destruction complex is mutated, then this complex doesn't work, and beta catenin will keep at a high level in our cells, leading to all the transcription factor will be activated, and all our genes will be transcribed when there is not even the presence of Wnt signal. So this will lead to cancer because our cells keep dividing, keep multiplying, and lead to cancers. So there's multiple reasons. Cancer can happen if the protein or the genes that produce Wnt is mutated, and Wnt keep be produced at a really high level. There's so many Wnt signaling, then leading to the cells will keep multiplying and lead to cancer. Yeah, so there are multiple reasons. Or maybe the mutant happen in crucial that make crucials not here, then lead to the transcription factor will be activated, and also in a high level of beta catenin, and this lead to the transcription of our genes also. So there's multiple reasons can lead to cancers if one of the protein along the pathway is mutated. Alright guys, thank you for watching the video, and I hope that helped or give you some information about Wnt pathway. Thank you for watching.